So this is the first day of our 5 days review. Remember, we're going to do this today, and we'll finish it on Monday because your test is Tuesday. Please read up there on the board. It even tells you. We even have a review session coming up this Saturday, too, if you guys want to come to that. So let's start things off with some important terminology. Always, everybody's most favorite thing in science is terms. So let's go ahead and define what science is. Science, what is it? Well, it's a systematic study of the world around us. I like this definition because I really like the word systematic. It means there's a process, there's a really a procedure-driven process that leads us to making discoveries by this study. We're looking at the natural phenomenon around us, the natural phenomena. We're trying to explain why things happen and what caused them to go down. But anyway, that's what science is. So then we have these things out there called non-science. I do not want you to associate non-science with something bad, quite frankly, because that doesn't mean it's bad. These are simply things that are not science. Okay, these include fields of study, still fields of study, like art. That's not a science. It's art. It's subjective. Science is more objective. Art is basically like, oh, I like that. And that's good. That's art. There are things out there like history. It's not as systematic as what we'd find in science because there's not a trial and error process. It's basically collecting as much information as you can and trying to put the pieces together to figure out what probably happened in the past. And then, of course, we have things like religion. Religion is not a science, but it's all cool. It's literally just a way of viewing the world, and it still can be a very, very good thing for this world, but it's just simply not a science. Now, there are some tricky ones out there that I want to warn you against are pseudoscience. Pseudoscience is basically, what does pseudo mean? Fake. Yeah, it means fake science. So basically, this is when people walk around and purport, hey, I have this cool scientific discovery, and it's going to change your life. Which I don't know how many times I've seen 90s infomercials that make these claims. But, like, you've probably seen, you know, people buy these, like, gold bracelets and put them on their wrists, and they're like, oh, it's going to give me better balance. My golf swing's going to get better. Sorry, I don't care if a famous golfer uses them. He's just a paid sponsor. That's cool. There's no scientific basis behind that. Have you guys seen, like, the clothing that has, like, copper interlaced in it? What? Yeah. What's that doing? I don't know. Scientifically, I can't explain what on earth it would actually be doing. It's not like it's a magnet. It's not like it's doing something for you, but they're like, oh, it enhances circulation, and more circulation is more oxygen, and you're going to do better. That sounds pretty fake. So those are things we want to keep an eye on. Things like astrology, homeopathy, and there's like dozens and dozens of others. Yeah. Sure. Though, I mean, once again, like, that's just tape. That's just tape. So can it fix a hole? Sure. You know what I mean? I wouldn't call that fake science. It's just like a dumb infomercial. Though he does slap that hole. Bang. Be pretty rad. Okay, so let's define biology. We don't have all the free time in the world here. So biology, it's going to be the study of life or living things and what affects it. Quite simply, we study things that aren't alive too in biology, like water and the atmosphere, because they would affect how things live. So that's important. Then we have this term, these two terms that kids always mess up: theory and law. I know theory might be part of your lexicon. You might use that phrase sometimes, but it doesn't mean what you think it means. Inigo Montoya. Anyway, so when it comes to theories are basically highly supported claims that haven't been disproven. So these highly supported claims basically means like somebody's made a claim, it's been tested over and over and over and over and over again, and nobody's been able to prove it wrong. So this actually brings up a key point in science, which is falsifiability. Basically, we want to see if we can poke a hole in your idea. That's what all science is trying to do. We subject our ideas to absolute torture, and if they come out and survive, well then by golly, we must have a pretty darn good idea. We're not saying it's proven, but what we're saying is it's been failed to be disproven. So it's the best idea we have now. Theories are literally the most complex and the most elaborately supported ideas humans have ever thought of. They're some of the greatest thoughts we've ever had. Because if they're going to make some crazy claim, they better have some crazy evidence. That's what a theory is. But a law is an accepted fact about how something works. We don't work with a lot of laws in biology, but there are things out there. Like there's the ideal gas law, or there's, oh my gosh, the law of independent assortment. Or, yeah, the laws of motion, which would affect gravity, right? We understand those things to be true. Nobody really argues those because we've tested this over and over and over again. It seems like these numbers all seem to work out. And then we use that to help explain other things. It helps us be more detailed. If we all didn't agree on the facts, then it would be really hard to communicate what discoveries there actually are in the world. So that's an important thing. Any questions here? Moving on. Let's do that scientific method. So the scientific method, there's a few blank spaces. Oh my gosh. There's a few blank spaces I gave you guys. Okay, all right. I'll change colors, make it fun. So what do we start off? Whenever we want to do science, what do we get right from the beginning, right from the get-go? What do you think?
A problem?
Yeah, we can start off with a problem. Some people might even phrase it as a question. Heck, some of you might even want to write down it's an observation. I don't care which three you choose, but basically you notice something and you want to know more. So once you have that, we move on to the next step, which is, yes? You need a hypothesis. No extra tiles for this one. Just to let you know, a hypothesis is a prediction we can actually test. I don't like the term, it's an educated guess, but that kind of has a nice thing about it, that you need to be educated, right? That means it's not out of nowhere. So I'm going to add an extra little loop here, which actually says we do research before we develop our hypothesis. That's super important to us because you don't want to just make things up. You want to actually base it in reality and actually understand if you're coming up with a hypothesis that's already been tested, then why would you test it again? So a hypothesis leads us to doing what? Yes, sir. Yeah, we got to design an actual experiment, right? Using that real hypothesis. After we design the experiment, by the way, the results of this would be variables and understanding what those variables are. After we design it, what do we do? Yeah. Yeah, you actually got to sit down and do that experiment. You have to do the thing you plan to do, because if you change it up, you may not be testing your hypothesis the right way. After that, it leads us to getting results. And those results, when we get them, when we think about them, we call that? Yeah, we got to analyze that data. There are different forms of data, both of which start with the letter Q, but we'll get to those here in just a minute. After we analyze our data, we form this thing called a conclusion. And that's always based on going back to our hypothesis, right? We always want to make sense of that hypothesis, don't we? And we can do one of two things. We can say that hypothesis is really supported or it's not supported. So if we feel like it is not supported, that means we go back and we come up with a new hypothesis and we start from scratch. Or maybe there's a chance you just like designed a bad experiment. There's even a possibility that you just did the experiment wrong. Well, whatever it is, you got to go back and think about what's going on. Review your work and find out if you need to improve somewhere. But if, let's say, we do support it, it leads us to this cool thing called peer review. Okay? Peer review is when you get together with other colleagues of the same field as you. You maybe publish your results, thank you. You publish your results in some kind of journal. They read it, they analyze it, and they determine whether or not it's good, whether or not it makes sense. And maybe they even run the test themselves and they confirm your ideas. If you do that, well then by golly, you might end up with a brand new discovery, which is pretty cool stuff. Any questions? You sure? No? Moving on? Okay. So let's go ahead and talk about some of those variables. We'd find this in our experimental design. We said there are 4 different parts. The IV, which is the independent variable, that's the one that's written on your little worksheet there. The independent variable, what does anybody remember what this is? Yeah.
It's the factor that you alter.
Yeah. I like that word altered. I'm also going to put in another word in there. Sometimes we use the word manipulate. And I just would point out, as a matter of fact, sometimes the independent variable is referred to as the manipulated variable. So I'm going to put that in parentheses next to it. This is the thing that we are playing around with and testing to find out if this causes a change, if this is really what causes it to happen, right? So if I change, you know, I don't know, what temperature I let plants grow in, right? And I have these different containers that I can change the temperature in, cold, you know, all right, cool, chilly, moderate, lukewarm, and then really hot. Like I can have all of these things in there, but I'm going to give them all the same factors, but I'm going to change the temperature. Well, then that's going to be my independent variable. My dependent variable will be the factor that is measured. Sometimes it's called the measured results or just the measured variable. But these are the end, you know, data points that we're going to look at and analyze later on. So we're looking at the dependent variables every time we do a data analysis. Then we have this cool thing called a control group. What's that about? What do you think?
It's a group that has no independent variable.
Good. Group missing the IV. And I'm also going to go ahead and mention here, we compare end results to this. This is super important. Even though really elaborate and complex like procedures have been done, and people get some cool data, and they're like, yeah, look, lasers. But if they don't have like a control group, then what are you comparing it to? How do you know if you actually made an impact on whatever it normally was? This is a big deal. And then we have these things called constants. What are constants? Yeah?
They're like things that never change.
There you go. Factors that remain the same. In the experimental design, we know that there's a lot of things that could influence the outcome, but we want to control as many of them, and the only thing we really want to change from set to set is going to be, of course, the independent variable. So my little experiment with the little containers with plants growing in there, we change the temperature. Well, I'm going to make sure they have the same quality of air, they have the same amount of water, they have the same amount of light that they receive. Everything's going to be the same except for the temperature that they're actually experiencing. So those would be our constants. Do you guys have any questions on those? You sure? Okay, last thing in this big box, we're going to talk about the different types of data. Super easy. Data comes in 2 forms. We've got quantitative. We've got qualitative. So quantitative, can somebody tell me the root word there? What do you think?
It's like how many there is.
Yeah, it's the amount, right? It's quantity, right? So it's an amount of something. It has a measured value. Basically, we're saying, oh, I can put a number on this, right? How long is this thing? How much did it grow? How much does the temperature change? I don't know, like what has the mass changed at all? All of those are factors that we can measure that way. Qualitative requires a description or characteristics. There's not a number for aggression, If you're looking at an animal and you're like trying to measure their behavior and they're aggressive, you can't be like, oh, he's a 3 out of 10 there, huh? Right? You can say, oh, well, they're very aggressive, they're very territorial. These are words that convey a thought and have a characteristic. You can't say, oh, it turned from red to blue. You can't use that as a number. You have to say, oh, it went from red to blue. So that's all qualitative stuff. Doing okay? Doing all right? Any questions? Groovy. So let's go ahead and talk about them atoms. Atomos. So let's go ahead and hit up the basics here. We've got protons, we've got neutrons, and we've got electrons. Hey, what kind of charge does a proton have? You guys are geniuses. What about a neutron? Yeah, no charge. And electrons have a negative charge. So let me go ahead and try to redraw the thing that's on your worksheet, which I drew myself by hand, but of course I can't do that again really fast and on the spot like this. But I tried. Oh boy, did I try. Let me change colors, make it a little easier here. So you're going to see on that inner shell, we call, oh no.
You caused it.
Ha ha, it's okay. So on that inner shell, I'm going to have 2 dots. These represent electrons that can fit in the electron cloud in something that we call an orbital. So the closest orbital, the second closest orbital, third closest orbital, and trust me, when you go into chemistry, they'll change all this stuff for you. This is called the Bohr model. But anyway, you'll see there's 2 dots there. That's the maximum number of dots it can hold. On the next layer, oh gosh, that's scary. On the next dot, we can hold 8 electrons. And on the last dot, I'm just going to get really lazy and write 8 there because it also holds 8 dots. Now, we only care about in biology these first 3 just because all the elements that we're going to work with that make up living things are probably within these first 2 rows of the periodic, or 3 rows of the periodic table. But anyway, on the inside, we have this thing called a nucleus. And that's where you're going to find protons and neutrons. That's where they are. And by the way, roughly speaking, the number of protons equals the number of electrons. So having this knowledge, I bring this up because it gives us a sense for how many extra electrons are on the outermost shell. Atoms don't like to have extra electrons on the outside with not a full shell. So what they'll either do is they'll gain electrons or they'll lose electrons so that their most innermost shell can now become exposed but full. But let's go ahead and talk about these things called types of bonds. So first off, we have these things called covalent bonds. Who knows anything about those? What do you know? Go ahead, sir.
It's like where they're like missing electrons, so what they do is they decide to like share.
Share. So it's the sharing.
Sharing is caring, baby.
It is. It is the sharing of electrons. I should also mention this is the strongest of bonds that we have out there. Next up, we've got ionic bonds. Oh boy, Ionic bond. What do we know about that?
It's like where one just kind of throw away the electron and another guy picks it up, and then that causes them like stick together.
Yeah, so if you gain or lose an electron, you become an ion, and ions are attracted to each other based on the kind of charge they have. Just knowing this, let's go ahead and hit up a couple examples. So a covalent bond, a good example, involves oxygen, right? Oxygen, I believe, is 8 on the periodic table, so that tells me there's 8 protons on the inside here. Yes, sir?
For covalent, it says sharing of what?
Electrons, sorry. So there's 8 protons in the center of our nucleus here. And then we're only going to work with the 2 shells here. Oh, I got to change colors. Here we go. Here we go. Calm down, everybody. So we've got 2, and then we've got 6 left over. 1, 2, 3, 4, 5, 6. There are 2 extra spaces that are found on this thing, and those extra spaces definitely need to get filled up. That's what wants to happen. So hydrogen, which is just 1 proton and 1 electron, it'll form a nice little relationship where they'll share the electron. So hydrogen's happy sometimes because it's got 2 on its outermost shell. Oxygen's happy sometimes because it's got an extra one on its outside of its shell. And typically what happens is another hydrogen sticks to it over here. This space right here is called a covalent bond. Then we've got ionic bonds. Ionic bonds are basically like this stuff called sodium chloride. You know that is salt, right? Well, sodium by itself is an ion. It's slightly positive because it's lost an electron, and chlorine is slightly negative because it's gained an electron. Well, since that one's slightly positive, one's slightly negative, they're attracted to each other, and they stick to each other, which creates the ionic bond. Last thing we have is a hydrogen bond. What do we know about that? Yes, sir. It is weak, extremely weak. That's true. What else do we know about it? What do you think?
It's like where the, it's like, no? So it's like where, okay. It's like where, the hydrogen of another, molecule attracts to, oxygen of another.
When hydrogen from one molecule attracts to oxygen of another. I couldn't have said it better. I will add in, it could be nitrogen as well, but nitrogen or oxygen. So overall, they have a negative charge, and that's that bonding that we have there. And we'll talk about that better on the next slide when I can draw this picture a little bit with more space. You guys have any questions here so far? Are you sure? Yeah.
You said hydrogen to nitrogen or oxygen. Is that because the positive and the negative together?
That's
So
exactly
hydrogen
it.
is just positive?
Yeah, as long as it's attached to a molecule, hydrogen probably has an overall positive charge, or at least that part of the atom, or I'm sorry, part of that molecule. And then it's going to find another molecule with another oxygen or nitrogen, and it's going to be attracted to that. So let's do WADAs in blue. Hey, look at me. Themes. Okay, so polarity. We've got a water molecule over here. And I've kind of drawn it, and on your paper it even has like this dotted line that runs through there. Looking at this, overall this side is more negative and this side is more positive. So the different charges on opposite sides of the same molecule. Okay, so like water has a negative and a positive side. That's polar, just like we have a north and south pole here on Earth. Think of it that way. They have two different poles. So some things you want to keep in mind here. Water does do bonds between other water molecules. So if I draw this up here, once again, this side's more negative, this side's more positive. This attraction that's found here, that is that hydrogen bond that we just defined. Because once again, it's the hydrogen of one molecule to the oxygen of another. So they kind of want to attach that way. And that's once again because of those positive and negative charges. So that is a hydrogen bond. And I should also just point out to you, hydrogen bonds are the cause for all the cool properties of water. All the weird stuff that they do, it's thanks to hydrogen bonds. So let's talk about those properties. We've got something called cohesion. Cohesion is going to be what? What you think?
Water to water.
Yeah. Water sticks to water. What about adhesion? Yes, sir.
Isn't that when water repels water?
No.
Oh.
Chris?
It sticks to other things.
Yeah, so when water sticks to other things. And then the next one up is high specific heat. I'm just going to get lazy and write high spec. Okay, what do you think that is?
It's that water doesn't change temperature very easily.
Yeah, it takes a lot of energy to change water temperature. And of course, the temperature of water defines what state the water is in. Is it frozen? Is it liquid? Is it a gas? And if you add a lot more energy to it, well, it starts to want to boil, right? But it takes a lot of energy for it to want to change that temperature. And also, you'd have to lose energy for it to drop temperature. This is really significant to living things because we're comprised of a lot of water. And if I reach my hand inside of a hot oven to pull out the cookie tray, well, then my hand's suddenly going to turn to steam. That sounds like a bad thing. So that's not what happens, right? You stick your arm in there. If it's in there briefly, all is well. High specific heat. Ah! Yeah, I yawned. Not because it's boring, but because I'm tired. Expansion when it's freezing. So basically when water expands, it means that the hydrogen bonds become spaced out. So what we're saying here is that the space between the water molecules is expanding. It's getting wider whenever water gets down to freezing temperatures. This actually causes ice to become less dense and causes it to float. It also acts as an insulator and prevents like bodies of water entirely from freezing because the ice will block the colder temperatures from making it deeper. Yes, sir?
Are hydrogen bonds found when there's effusion?
Yeah. Hydrogen bonds are literally the reason why the water will stick to something other than water. A good example is like in capillary action when we talked about plants, right? We said they kind of draw up water through their roots. Well, the roots have like an attractiveness to the water. So the hydrogen bonds will form with the root. And the cool thing is behind that water molecule, it'll probably pull other water molecules with it. And that's why so much water moves in the plants, because of those hydrogen bonds. So we've also got solvency, which is the word I used there. I'm not sure if I'm using it right. But basically, if there was a lead contender for a universal solvent in the world, it would be water because it dissolves most things. Okay? And when we're saying it dissolves most things, we're saying like literally it's doing that. It's trying to break those things down. It doesn't mean it'll break everything down quickly or even well at all, but at least if anything's running for contention, this would be it. So, do you guys have any questions on properties of water? What? Oh, I thought you said brochure, and I was like, okay, I'm not handing out any pamphlets today, but... Okay, so when it comes to these molecules, which is the worst spelling of molecules on Earth, we have these things called monomers and polymers. Hey, mono means what? One. What does poly mean? Many. So what we're saying here is to form a molecule, we need some small components, and that's what these are. Small components of larger objects. Basically what we're saying is you got to get like special kinds of atoms that link together maybe more simple molecules, and they together form more complex molecules, and that's what a polymer is a complex molecule. So like on a goofy example, like on bricks on a wall, right? This is the worst brick wall ever drawn on Earth. Whatever, it doesn't matter. Each individual brick is a monomer, right? But the whole thing together is a polymer, right? You with me? You remember that? Okay, it's like building a train. Each individual train car is one monomer, but when you put it all together, we start calling that thing a train. And it starts transporting things, and people jump on it and travel through the desert. I have a little knapsack with them. Anyway, moving on. Can we flip the page? Wow, we're 1/10 of the way done with our entire review, guys. I know, right? Okay, so we've got a really... What's that?
I missed it already.
Oh, I do too. But don't worry, it's just on the other side.
How are we doing on time?
We're doing all right. But let's see. So, you guys have this chart that's in the upper left-hand corner of your second page here. I'm going to go ahead and like kind of set up one. I'm just going to say contains. I'm going to say monomer. I'm going to say function. And I'm going to say example. Okay? So our first thing are carbohydrates, right? So what do they contain? And we're saying what atoms do they contain? Well, there's only a few options here, but it contains carbon, hydrogen, and oxygen. Those are the only three things they'll contain. The monomer that they are, it's made up of these things called sugars and starches. Sugars are usually their monomer. The most basic monomer is called a monosaccharide. Saccharide just means sugar. One sugar. There are disaccharides. There's trisaccharides. By golly, there's even polysaccharides, which obviously are the more complex of the things we've listed there. So their function, well, they do a lot for us, but what do we mostly use them for? They're the main energy source, which I heard some phrases out there, so I'm going to go ahead and write down. It's short term. Okay, like if you ever drink a soda and you're like, and you get all excited, because that's a really simple sugar, and your body's like, hey, I can digest that pretty quick, or at least most of us can, and then that gives you a little boost of energy. So it's a main energy source, but also for plants. It's used for their cell wall, so it provides structure for plants. That's called cellulose, by the way. It's the fiber in your diet in most cases. It helps you go poopy poop. Anyway, examples include things like, hopefully you've heard of this stuff before, glucose. Yeah? Glucose. Yeah. C6H12O6. It's a really simple sugar, pretty common. There's also something out there, I think I already mentioned it, fructose, like high fructose corn syrup, which basically is pulled from veggies and some fruits. Who? Good for them. And then another one is glycogen. Just a little bit more fun information. Glycogen's a sugar that's stored in your muscles that is processed by your liver. If you ever do a prolonged physical activity and you feel tiredness in your muscles and that soreness, a lot of the times you've actually lost a lot of your glycogen, which is pretty bad. You should always try to replenish those things. Anyway, so we got carbohydrates. What about lipids? Yeah? They're comprised of only two kinds of atoms, carbon and hydrogen. The reason for this is because they form these things called hydrocarbon chains. They're really long and complex, and eventually what happens is when they're broken, they release a lot of energy. So they're monomer, which they really don't have one, but if they did, it would be the fatty acid. But they also have this cool thing called glycerin. Or, I'm sorry, glycerol. Glycerol is basically the thing that attaches and holds the tails kind of together so they don't just kind of break off and float around wherever they want. Their function is what? No, we've already got short-term energy. Yeah, there we go. Long-term energy. Basically, it's stored on the body. And also, they do some other cool things. They make up the cell membrane. Remember the phospholipid bilayer? No? Well, phospholipids are lipids, and they make up a big component of cell membranes. And basically, that's a lot of what they do. I mean, that's already a ton of things. But there are different versions out there. I should just tell you there are things out there called steroids. Not all steroids are like, I need to gas up, brother. I mean, like, steroids meaning, like, they're chemical signals that are created in the body. And sometimes they use them to treat people that are just sick. They might put you on steroids just to enhance your ability to recover. Good examples of these things include things that you're all familiar with, like fats, oils, and of course, waxes. Like earwax is actually a lipid. Isn't that fun? I know. Protein. Protein is made up of carbon, hydrogen, oxygen, and even nitrogen. That's an N, sorry. Their monomer is amino acids. I see that some people finally got the tattoos that I told you to get. Hey, you still got one more weekend to contact your tattoo artist and get a tattoo that says proteins are made up of amino acids, or amino acids make up proteins. But their functions, while they are numerous, let's try to limit it down just a little bit. They regulate cell processes. They do some cool stuff, like they make up bone and muscle. So that's when you hit the gym, what you're doing is you're like breaking down some protein structures in the muscle, and you need to make sure you have protein in your diet to replace that, because you got to fix it later on. Transportation, cell transport. They're used for that, and they're even used for identification. You guys remember talking about the immune system? And we said like different types of blood are out there, and we said there's little identifiers, or there's little like antigens or antibodies that are found on the outside of some pathogens. Those are all proteins. Good examples of these include things called enzymes, which we'll talk about here very shortly. The last thing we have are called nucleic acids. They're made up of all of it, C, H, O, N, and a fun one called phosphorus. Yeah, they're made up of it all, and even sulfur. But anyway, their monomer is made up of these things called nucleotides. Hopefully you remember those nucleotides because we talked about adenine, guanine, cytosine, thymine, and even uracil in some cases. What is this used for? Genetic info. That's it. That's all its purposes. It might help in the processing of making some proteins, but it's still a form of genetic information that actually helps code for it. Good examples of these include DNA and RNA, DNA and RNA, right? So those are everything that you should basically have in your little table-y chart-looking things. How you guys doing? Yeah, I feel you. I've been doing this a lot today. So, no questions there? Okay, so let's move on. Hit up the enzymes, y'all. They are exciting. Remember doing the enzyme lab? They help things to go? Yes, it's just like ordering to go at any fast food restaurant. You get enzymes, which is actually more true than you might think it is. But anyway, let's go ahead and look at what a catalyst is. A catalyst is going to be a substance that speeds up chemical reactions by lowering something called activation energy. Okay? So activation energy, you're like, what? Huh? What? Activation energy, I'm going to define it right here. It's the amount of energy needed to start a chemical reaction. Okay? So to make more sense of this, when we look at our little graph that I gave you, oh yeah, phones are ringing. It roughly looks like this, right? And hopefully you're noticing this, but maybe I made it too small. I gave you some like blocks to show us that things are combining from being separate. Anyway, let me change colors. You'll see. Oh boy, that's embarrassingly bad. I apologize. The red line is when you're using an enzyme. The blue line is when you're not. And you'll notice this distance from up here to here, that is what we call the activation energy. That means you need to get this much energy before your chemical reaction can even take place. Well, if we use the red line, it's much less energy required, which would save us energy, which would mean we can probably use that energy to repeat this process, which probably means we're doing it more often. So that's the main relationship between those things. I should also point out to you, they can be affected by a few things. They can be affected by temperature changes. Enzymes are proteins, right? So if you change the temperature of a protein dramatically and make it too cold or too hot, you denature the protein, which means it unravels and falls apart, which is not good for the protein. Well, it's not a living thing, but yeah, it basically dies. It cannot be repaired at this point. Of course, we can change the pH of these things. If we make it too acidic or too alkaline, that could make them fall apart. We can also change the salt concentration or even just overall concentration of the enzyme itself. Okay? This will affect the structure of the enzyme and cause it to fall apart. But with concentration, just the number of enzymes there will affect your enzyme activity. If you don't have enough enzymes, it doesn't matter. You're not going to be able to do this fast enough to actually make a significant improvement on reaction times. I'm running a little short on time, but let's see if we can hit this up. Lock and key model. And basically, enzymes are like big, ugly things. Yeah? And there's pieces that are going to fit to them. Wow. That's really terrible. Yeah, it is. I guess so. But this is the enzyme. And this is what we call a substrate and a substrate. Well, they got to fit together. And there's actually like special parts that fit here for the enzyme too. Since it fits together like a key going into a lock, they only work with certain enzymes. So that's why somebody who's lactose intolerant and can't break down sugar from milk, lactose, if they don't, they're missing this enzyme called lactase, so they can't break that down. But that doesn't mean they can't break down anything. They can still process things. They still have amylase. They still have catalase. They can still break down other parts in their body. They just can't process this particular thing because they're missing that particular enzyme, which is the main concept of the lock and key model. Even though it's a bit outdated and old-fashioned, because nowadays we really know the truth is it's more like the induced fit model. But let's not worry about that and save that for another class like AP Biology. So you guys, you good? You ready to move on?
Yeah.
Okay. We're doing great on time now. pH. Keeping this as simple as possible, we have two things. We have acids and we have bases. Okay? Sometimes people use the phrase alkaline to describe a base. But acids and bases, they're set up on the scale. Attempted to draw a straight line. Yay! Okay, so the scale roughly goes from 0 to 14. In the middle is 7. And I should just point out to you, pure water, absolutely pure water is exactly middle of the scale, which means it has this equal balance of being an acid and a base, so it's not very reactive. I'm just going to point out to you, acids are basically considered having high amounts or high hydrogen proton amounts. Honestly, it's just a proton that's floating around that fell apart from water, essentially. And bases are known for having low amounts of hydrogen ions. There's actually two different scales. There's another scale called POH, but hey, whatever. We're not worried about it. As we move further away from the 7 in either direction, we go from being a weak thing to a strong thing. So if you're closer to 7 on both sides, you're either a weak acid, which this is the acid side and this is the base side. If you get closer to 0, you become a strong acid. And if you get closer to 14, you become a strong base. Just try to remember, acids are closer to 0, bases are closer to 14, 7 is closer to pure water, which means it's rather neutral. So you're weaker the closer to the middle you are, and you're stronger the more to the extremes you are. Remember, good examples of acids includes things like hydrochloric acid or sulfuric acid or battery acid, you know, the stuff that can eat through your skin. And then we've got, on the 14 side of things, we've got like a lot of cleaners, like ammonia, bleach. We also got like, what's the super strong one? Oh yeah, drain cleaner. All of those are really, really strong. Things closer to 7, yeah, we'll have a range where life likes to live. And you've heard of acid rain, so acid rain's a little more acidic. You've heard of salt water, which is a little more basic. Hey, all right, cool. Baking soda, stuff like that. Doing okay?
Yeah.
Okay. Cell theory. Yay. 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 Let's go. Let's do this. There are 3. Oh, there's this dude named Robert Hooke, right?
Yeah.
What did he do?
I don't know. He was just like, hey.
Yep, he discovered cells, and he was like, hey, I'm going to write down discoverer. Anyway, there are 3 postulates to cell theory. This was developed by a lot of people over a long period of time, including not only the work of Robert Hooke, but guys like Anton von Leeuwenhoek, Schleiden, Schwann, kind of crazy names. But anyway, there are 3 postulates. There are 3. What are they? I love it. All cells come from other living cells. Really important, because a living thing can make a living thing. A dead thing does not make a living thing. That was a big discovery. Disproved like kind of some concepts about what germs were, or what made people sick, and even like where life came from. We've got two others. What do you think?
The cell
all of
is smallest unit.
Okay, I was calling on him, but
Sorry.
all right. The cell is the smallest unit of life. I agree. And then,
all living things come from other living things.
well, I think we've got to,
I'm sorry.
yeah.
I'm sorry. All living things come from cell are made of cells.
Okay, there we go. All living things
Messed up a bit.
are made of cells. So those are the three main postulates. Did I put them in the wrong order? Yeah, probably, but it doesn't matter. You got to know these three concepts in terms of what cell theory is. Once again, just to point out to you, cell theory is a theory. Doesn't mean it's a weak idea. Not at all. What it actually means is it's one of the most brilliant ideas we've ever had, and people don't debate whether or not we're made of cells, even though that's a thing. Finally, we have microscopes. And of course, microscopes, well, they've played a really big role in, science in general. But what we want to know is, how do they actually work? Of course, they are a series of shaped lenses. And they're basically shaped to cause magnification. So we can observe objects a little bit more closely. There are, of course, pieces and parts that really matter to us. There's the coarse adjustment knob. There's the fine adjustment knob. Of course, the coarse one is the big one. The fine adjustment is the small one. And when you turn these, they move the stage closer or further away from the objective lenses. The objective lenses are the lenses that are closest to the object you're looking at. And basically, whenever you kind of twist that, you can change the magnification of those lenses. And then, of course, we have the ocular lens, or the eyepiece. Actually, if you multiply those by each other, those two numbers for those lenses, you actually get the total magnification. And then, of course, we have the other parts like the arm, the base, the light, because these are light microscopes that we used. And we even talked about other kinds of microscopes that are out there, like scanning electron microscopes or transmission microscopes. Scanning electron do a really good job on giving us high resolution, and we look at the surface of objects, whereas transmission kind of looks through those objects, and we can see really, really tiny things like inside of cells, and we can distinguish cool stuff like that. And the other question is, how did this change the world? What change was created from this? Well, basically, it led to the discovery of cells. And that discovery of cells, of course, prompted a whole lot of other discoveries. Of course, we developed cell theory, but of course, we helped